Assalamualaikum and good day everyone. I will be presenting on my final year project on development of aerobic granular sludge using low strength domestic wastewater. Let's move to introduction. Growth rises in urbanization will inevitably require the development of new water resources, leading to an increase in the amount of wastewater which must be treated before it can be released into the natural ecosystem. Generally, wastewater treatment includes four basic components, which is physical, mechanical, biological, and chemical. Aerobic granular sludge is one of the wastewater treatment systems. The conventional activated sludge process system is the standard technology for biological wastewater treatment. The AGS technology has been developed in and is currently mainly used to treat urban wastewater with high concentrations of organic compounds, which is chemical oxygen, which mainly come from different sewage systems. The activated sludge method is currently the most widely used biological method in domestic wastewater treatment plants because bacteria and other forms of microorganisms in the system need oxygen to survive, expand and multiply in order to absorb this organic matter or contaminants in the wastewater. In this treatment method, the wastewater that contains matter organic dissolved in the aeration tank will be gas. So, for the problem statement, the aerobic granular sludge process is a new method for treating domestic and industrial wastewater. Self-immobilized bacterial cells for solid, compact, and well-settling aggregates in granular sludge, which can be thought of as a special case of biofilm. Because of the granules properties, the process can accumulate high loading rates as well as radioactive substance in a small bioreactor, with rising pollutant loads and often limited capacity to expand existing treatment plants. As a result of urbanization, this effective low footprint process becomes an appealing alternative. Despite numerous studies on the use of aerobic granulation technology in the treatment of domestic wastewater, research on low-medium strength wastewater appears to be lacking due to low and complex substrate availability, which does not favor aerobic granule development. The aim of this analysis is to see how aerobic granular sludge develops in low-strength wastewater. At the Indah Water Research Centre in Kuala Lumpur, a sample of domestic wastewater is taken and the experiment is carried out. The purpose of this proposed research is to study the formation of aerobic granular sludge in low-strength domestic wastewater. Therefore, the specific research objectives will be as follows: to prepare and determine the characteristics of raw wastewater sample from extended aeration plant located at Bonus Indah Water. The second is to characterize the conventional sludge that has been used as a seeding to start up the AGS bioreactor. And the third is to monitor the formation of aerobic granular sludge in the AGS bioreactor. For the scope objectives, the aim of this research is to see whether AGS can be grown in a pilot-scale bioreactor using low-strength domestic wastewater. The following scope must be accomplished in order to meet the study's goal and objectives: the characterization of domestic wastewater and seeding sludge, as well as the growth of AGS, are the two main components of this research. Indah Water Research Centre supply the wastewater and seeding sludge. The bioreactor is housed at Indah Water Research Centre and the experiment is also held there. Sequencing batch reactor was used to build and run the bioreactor. The bioreactor was designed to run continuously for 24 hours, with each batch completing a 3-hour cycle. The wastewater sample was taken and the return sludge would be used to make new activate sludge. The ink column from the freshly returned sludge will then be used to start the granulation process in the bioreactor. Next, for the methodology for the study online, the experimental work for this study focused on the development of AGS by using low-strength domestic wastewater in pilot-scale bioreactor for treating domestic wastewater. This This chapter describes the pilot-scale experimental setup and analytical procedure in order to determine the formation of aerobic granular sludge using low-strength domestic wastewater. The parameters involve the concentration of chemical oxygen demand, total inorganic nitrogen, ammoniacal nitrogen, nitrate nitrogen, phosphate, biological oxygen demand, and total suspended solid. A pilot-scale sequencing batch reactor was set up for AGS studies at Water bonus STP, which was operated at ambient temperature about 27 until 30 degree. The development of AGS in pilot scale sequencing batch reactor was monitored from time to time. Material preparation for domestic wastewater. In the Water Research Centre, a local municipal wastewater treatment facility provides a sample of pure domestic wastewater before any form of treatment with a mesh of 1 mm to remove large debris and solid materials that can clog bioreactor influent tubes. 3 times a week, the sample was collected and fed into a bioreactor device. The wastewater characteristic analysis was also performed on the same wastewater. Next, for the seeding sludge, seed sludge samples from the IWRC aeration stand were used to make the perfect seeding sludge. 1.125 liter of activated sludge samples were planted in the bioreactor at the startup stage. The seed sludge was saved with a mesh of 1.0 mm to remove large debris and inner impurities. It was aerated for 24 hours before being continuously fed synthetic wastewater. The biomass increased after a period of decay, indicating the microorganism has adapted to the new feed and were ready to move on to the next stage of the experiment. The seed sludge had a loose, fluffy structure and was brown in color. Next, for the bioreactor setup, the experiment was performed at the Indah Water Research Centre in Wangsa, Kuala Lumpur. The working volume of the CAGS Bio, which is cyclic aerobic granular sludge bioreactor, was 2.5 liter. The bioreactor was of the column type with a tall and slender form. The figure 3.1 shows a 2.5 liter cyclic aerobic granular sludge in the laboratory environment. During the startup time, 45% of total volume of the bioreactor was filled with written sludge from IWRC assay columns. Digital timers configured according to the required duration operate the feeding pump, discharge pump, and aerate pump for each step in the bioreactor. Then the bioreactor was designed to run continuously for 24 hours with a total cycle time of 3 hours. Then the process begins with the 60 minutes of anaerobic feeding from the bioreactor's bottom without stirring, followed by 84 minutes of aeration, 30 minutes of settling, and 6 minutes of effluent withdrawal in one period. Next, for the wastewater characteristic analysis, the organic nutrient, inorganic compounds, and toxins can all be found in domestic wastewater. pH, chemical oxygen demand, biological oxygen demand, phosphate, ammoniacal nitrogen, nitrite, nitrogen, nitrate, total nitrogen, total suspended solid, volatile suspended solid, and total dissolved solid were all used to characterize water in the water. The parameter involved in the wastewater characteristic all will be tested. The stated is the procedure of pH, COD analysis, total phosphate analysis. For the seed sludge characteristic analysis, using seeding sludge from the written sludge, and an aerobic granular sludge was developed. In an effort to provide an optimal seeding sludge, sewage activated sludge was used. It was also used to develop aerobic granular sludge as a source of microorganisms. The properties of freshly activated sludge was investigated in this analysis. MLSS, which is mixed liquor suspended solids, mixed liquor volatile suspended solids, sludge volume index, size, and certain velocity were all used to determine the physical characteristic. As shown in the table, this is the result for the characteristic of domestic wastewater. which include all parameters which is pH, alkalinity, COD, soluble COD, BOD, ammoniacal nitrogen, nitrite, nitrate, total nitrogen, phosphate, total phosphorus, total suspended solids, volatile suspended solids, total dissolved solids. Then also the results of physical characteristics of sludge, which include parameters of pH, MLSS, MLVSS, SVI, size, and cycling velocity. Okay, next for figure 4.0 and 4.1 illustrate of COD in influent and MLSS in bioreactor. As can be seen in the figure, the influent of COD concentration range from 260 mg per L to 316 mg per L. The performance of SPR in COD removal is dependent on the availability of oxygen within the system. The higher COD indicates the higher amount of pollution in the water. The range MLSS in bioreactor from 5,465 mg per L to 3,880 mg per L. Lastly, in conclusion, aerobic granulation has been demonstrated as a promising treatment alternative for actual low-strength domestic wastewater treatment based on carried out at lab, pilot, and full-scale levels. Aerobic granular sludge was successfully developed in short time in pilot-scale bioreactor with real domestic wastewater from indoor water bodies at ambient for 27 degrees Celsius. The formation of aerobic granular sludge can be developed rapidly with the low-strength domestic wastewater. That's all. Thank you.